Okay, let's see, what do we got? Okay, we got 10 o'clock up and down. Okay, so I'd like to remind everybody that this is the information about the panel. We've got 3 panelists, and this is a look back at some of the events that we had in 2020, which, as everybody recalls, was the COVID time. So we're, what else could you do? So we're very prolific in presenting stuff. So the way we're going to go today, hi Ellen. So the way we're going to go today is we've got 3. I'm going to kick it off. And then it's actually going to be Dr. Stephen Gasior's second. And then Dr. Linda Morris Kelley will finish up. She's at another event right at the moment, and she'll finish up in the last 20 minutes with her presentation, a little field trip around here. It'll be a good experience. Okay, so let me go ahead and kick this off. There you go. Everybody get a seat in the grass or wherever it is. And I thought it appropriate for this area to kind of tilt my presentation. So let me get going. Time is very interesting. I have always been interested in time, and I did a presentation on time back in 2020. Time is one of the things that depending on how you define it, it's one of the things that every object is affected by. Every object basically changes. You can't you can't get rid of it politically. You can't affect it economically. It's basically here for everybody in equal ways, and it's timeless, etc., etc. So let's take a look at a little bit of the both psychology and physics, so to speak, or science of time. So in our consciousness, time can be many things. It can be a fixed time marches on. Just a second. Very defined units. Oh yeah. Sorry. Okay. Like what Stephen said. In other words, I have the first 20 minutes. But what I'd really like is for people to comment in chat as we go, and comment, ask questions, links, the whole bit. In other words, a dynamic get together here. Okay. Well, yeah, Ted. Light. Maybe the one independent thing, so to speak, although, light travels at different speeds in different media. So I'm not so sure. It's not a physics lecture necessarily, but it has to do with both our perception of it and how we measure it and stuff like that. So, is it fixed? Is it relative? It all depends. If we were to, just sit here and there was nothing going on, it would seem like time had stopped. But we know very well that inside us, our cells, everything is going crazy inside of us, and all the little atoms and all that. So, and they're not affected by what we think. So time can go very fast. If you're in a fire like here, time can slow down. Time can seem endless, and yet it can also, if you're just tranquil and looking at things, time can be slowed down. So time can also be, exactly, seem endless. Time flies when you're having fun, or when you're older. I gotta tell you, I'm mid-70s, so time seems to go a lot faster than when I was younger. Okay, so anyway, time can also appear linear. It can appear cyclical. Is it real? Is it fruit fly like a banana? I love it, Sis. Okay, so, Sis, of course, is our local Loki, jokester. And time, is it real? Is it construct? Is it, imagination? So this presentation, what I've done is taken some slides from the presentation. Oh yeah, thank you. um, from the presentation back in 2020. And in Western cultures, we tend to think of time. Well, okay, that's generalizing a lot. Western cultures can be lots of different people, okay? But in cultures that originated in Europe and such, we or people in that culture identify with the culture tend to look at time kind of in the arrow of time, time marching on, time as being progress, time as being end times at some point, that sort of thing. But in other cultures, back in 2020, it was the year of the rat, and in Asian cultures and in lots of places in the world, time is not an arrow, it's a cycle. There's patterns that repeat, not exactly, yeah, time, rosemary, and whatever. Um, so patterns that repeat over many, many years, many generations, many cycles of birth and growth and death and rebirth. So some of the emphasis there in red, I did that as kind of an update for this time, since I only have 20 minutes, is you'll notice what's fascinating when you study time is that a lot of it seemed to have been, some of the ideas about time may have existed long, long, long before any of our history. There is a story in the Native Americans in Northwest California area about two gods represented by the volcanoes, Mount Mazama and Mount Shasta, fighting each other, and then the Mount Shasta god won, and Mount Mazama collapsed. And that actually is a real event that took place 12,000 years ago. Mount Mazama is what we now know as like Crater Lake in Oregon. And so some of those stories may have existed many, many thousands of years and then been passed on. So you have this idea, if you look up there at the philosophy of time, you have in the square red, both in the Vedas, in the Hindu scripture, and in, yeah, it is, a very interesting mist. And then also in the Mayans, in other words, half a world around, you have the same idea of 4 cycles, 4 world ages. If you look at the Mayan creativity or creative story where it says, does any of this sound familiar? Where all is stillness and water, and there's a separation, and then these brothers separated the sky and earth, and they're made an underworld and an upper world of different levels, and created humans from mud. Does any of that sound familiar? Like as in Near East, creation stories, even though this is the Mayan. In some, yeah, cyclical time. There's no future, that sort of thing. Sometimes you don't even have words for that. I'm going to be kind of roaring through these slides a bit because this is not supposed to be an hour-long presentation. I have 20 minutes. Okay, so, but sometimes when we look at how time is presented, when we try to measure things like the heavens and stuff, we can then not progress if you think about it. In other words, for 1,000 years, we've progressed some of the ancient errors about our thinking of the solar system, where we said, people ask each other, did you create Venus? No, I didn't create Venus. Okay, so therefore it must have been created by a superior being, and it must be perfect, and there must be a round orbit, and all these things that then took 1,000 years or more for somebody to go, hey, by the way, if you actually measure things, it's not perfect, it's not a perfect sphere, etc., etc. Okay, so when I was, when I was young, and people go, time is money. Okay, if you're particularly in the West, and you think of productivity as the measure of your being, then in that case, time is money. In other words, if you're not going rush, and making as much money as possible, then that's what time is money. But when I was in college and didn't have any money, I was thinking, well, what is money? And I came to the conclusion that money is time. In other words, if you had time to grow vegetables, time to make a car, time to learn how to make a car, that's great. But basically what we do is we get money and we pay for somebody else's time to have made that car, made the food, whatever. So I came to the conclusion it's not just time is money, but money is time. There's other ways we look at stuff. And so when we say life expectancy, we can't really go, okay, great, I'm going to live to 76 and die. It doesn't work that way. It's a probabilistic thing where essentially for people that were born around my time, that 80% of people will live past 57, but 20% will not. Steve Jobs, for example, died at 58. Average person might live to 68, which I remember well. But then the mode will die at 77, which means not everybody kicks off at 77. But you get what I'm talking about. In other words, when you talk about time, you kind of have to define it carefully. Now, that's kind of the philosophy of time. If you look at it from a cosmos standpoint, the actual scientific or physical basis of time, it has to do with patterns. Where we see orbit, it's like, oh yeah, okay, the moon is in this spot of the sky, and then it's come back to this part of the sky, and that must be a month, and that kind of stuff. Okay, so there are natural cycles of plants and animals, which of course is why we invented calendars and stuff in the first place. So we knew when to plant crops, when to harvest crops, when birds would be there, when it would be cold, when it would be hot, all that. Now, when time starts getting messed with, like as in climate change, there's an example in red of what happens. So with warmer temperatures, the plants get leaves earlier, and the insects emerge sooner because they then eat the leaves. Well, the problem then is birds that are migrating, say down to Africa or South America or someplace, arrive late, and all the insects are now turned into, say, moths or butterflies, so there's less food and their little babies starve, and populations decline. In the case of birds, in North America, there's been a 50% decline in the last 50 years of birds, which is quite alarming. There are also clocks, time within us, circadian rhythms. If you take a look at this is internal time, and you can see our deepest sleep then is 2 o'clock, and our highest blood pressure is around 6:30 in the evening, and our best coordination is at 2:30, and all that good stuff like that. You can see how all of that works together, and it's surprisingly similar, as in science circle. Okay, but when you start looking at the Earth, it's anything but constant. It's anything but perfect. It wobbles. The day is not exactly 24 hours, nor the year. The Earth varies in eccentricity and obliquity, and it's just so you have to define, if you actually look at, well, how long is a year. So there's nothing at all that's constant with regards to the Earth, Moon, Sun, or anything else. It's all changing. It's just over very long periods of time, in terms of hundreds of thousands or millions of years. So if you're really going to say, if you look at the bottom, what I put, 265? I'll go back and look at it. I'll go back and look at that. Okay, so anyway, precise measurements of time is, long ago it was like, okay, it's lunchtime, great, okay, and that's all you need to know. But now if you look down at the bottom, if you're actually trying to coordinate satellite events, you've got to have very, very precise measurements of time that are based not on things like the moon and sun, which have no constant anything, but on atomic wavelengths, that sort of thing. Okay, now if we actually go out in space and we start living on Mars or the Moon, then we've got to think in an entirely different way, because now most of the things that we measured down on Earth, even gravity, which affects time, will be totally different. So we need to think about. In fact, right now they have been thinking about how to measure time on the Moon. So now, okay, we've looked at the kind of philosophy and how time is measured physically. Um, but now let's take a look at calendars. I've always loved, like I said, I've loved time and the idea, okay, calendars. And you have linear calendars and you have cyclical calendars. And they basically arose, the idea of fixing time was so that we could coordinate events socially. There's a lot of calendars still in effect worldwide. You've got the Hijri, and if I pronounce that wrong, let me know, calendar, and the Gregorian one, which is the one that is used for a lot of economy. But you also have indigenous calendars, and you have ones that are local calendars that people have been using for a very long time. And then even when you look at days of the week, they can change. And when you look at how we got our names for, this is just the European version here, English and Latin-based languages. You've got Latin and Germanic and Greek and Roman and all kinds of different mythologies working in there. And so if you look at calendar differences, a lot of people can't even agree on what day of the week it is. Now, the international ISO standard says it's Monday. Okay, but in some places it's Sunday. In other places it's Saturday. In other places it's Friday. Even the length of the week. The Romans had an 8-day calendar, an 8-day week. French Revolution, they tried to do a 10-day week. And then the Soviets tried to do a 5-day. And then the other... So it gets very interesting if you look around the world. Here again, I'm running out of time a little bit for this. I've got a few minutes. 8 days a week. Ha, ha, ha. Very good, Tig. Okay. Now, some of these you can actually, they don't say why, but if you really wanted to like calculate Easter, that's how you calculate Easter down at the bottom. Yeah. And then there are events like big earthquakes and tsunamis that actually change the length of the Earth's rotation, believe it or not, by like microseconds. Okay, now these ones, you will see calendars like this, where there's a cycle within a cycle, both in the Chinese Asian type calendars as well as the Mayans. As I mentioned, even though they may not have known about each other, you have these same cycles within cycles of time, sometimes going back millions of years before Europeans even thought about millions of years in time. And now I'm up to 8 minutes in, and that's the quick and dirty recap of the presentation I gave in 2020. And does anyone have any questions before I turn it over to Stephen? You can speak in voice if you want, or in chat.
Well, I have a question for you, Phil.
Sure.
In revisiting your video and what you just showed here, it's very interesting that a year is very well solidified to be one rotation of the Earth around the sun. But why is there so much leeway and play in the number of cycles you have? Like how many years per cycle or how many days per week? Why is that just because there's just no anchor to believe how long one of those cycles or a segment of time should be?
Well, that's a good point, is that the Earth going around the sun was a very real thing if you happen to have seasons. Now, of course, even that, it depends on whether you say, okay, has it gone around once, or has it gone around with relationship to the stars, or has it gone, in other words, there's tropical year and sidereal year and et cetera, that are all a little bit different. And there you go, like, grounded in moment. Now the moon, same thing, although since we are also going around the sun at the same time, how long a moon day is depends on what it's related to. Is it related to going around and then passing the sun? But there's a certain, number of degrees that are different, which change it from anything from 27 to 28 to 29 days, depending on what you're measuring it from. But when you're saying like how many days in an increment of social time, then it becomes more difficult. A lot of calendars, and now I'm up to my 20 minutes, but a lot of calendars didn't even bother with winter, for example, because they were mostly agricultural, or they were religious or festivals and stuff like that. So they didn't bother with nice
Hey, Phil,
increments. Yeah.
Phil, may I jump in here? I mean, Stephen's question is an interesting one about cycles.
Yes.
The thing is, as you said, as Roy said in the chat, I mean, it also has to do with how the moon revolves around the Earth. That's another cycle. But the Mayans, for example, which you mentioned, they were for some reason they were really obsessed with the cycles of Venus, seeing Venus in the morning and then in the evening, because it goes from one side of the sun to the other faster than the Earth does. So they were timing. They had a calendar that was based on how Venus moved in the sky. They also related that to how the year changed, how Earth revolved around the sun as well. So you can get into all kinds of different cycles and how they mesh with each other.
Yeah, it's very fascinating. That's why I like the whole topic. The Mayans, like you said in particular, had two. They had two cycles that meshed with each other and then came back together over a certain period of time. I don't have the slides available right now, but if you really want them, I can send them to you. You just have to get a hold of me, and I'll fix it up so that you can have them. Okay, here again, we could talk about this whole thing for an entire hour, but I will. Yes, yeah, a very complex society and calendars, which, by the way, Mayans still follow in Guatemala. Okay. Okay, there you go. Okay, so I'm going to be quiet and I'm going to let Stephen jump in here.
All right, wonderful. Thank you, Phil. One thing I want to provide to everyone is a listing of all the videos that we're revisiting from the talks in 2020, because it was quite an interesting year. But on top of that, Science Circle was very active, had a lot of great talks on different items, and we actually hope maybe we can keep revisiting it. So the talk that I gave was a little bit personal, but also trying to talk about what I think will be an interesting topic going forward in terms of health, human condition, and potentially a Nobel Prize for somebody. So I talked about genomic parasites nibbling away at us, and really the basics of the talk were, I published in this field, and the basic idea is that there are these things that people call colloquially junk DNA. They jump around our genomes. They're kind of parasitic in that they're primarily interested in their own replication. And my specific contribution was to say that it makes something called a double-strand break. And again, this is thinking about if you recall that the DNA helix is 2 strands, if you make a break all the way through that, that's what we call a double-strand break. And in my work, I also showed that genes that we know are involved in repairing double-strand breaks also seem to play a role in either keeping these things from doing that type of stuff or actually help aid it. They're enablers for these things to do their thing. And the talk also included some other background, which was this idea that DNA repair deficiencies, when they are within a human or different model organisms, can show symptoms of premature aging. There are these things called RAD genes, which were found because they created sensitivity to radiation, so X-rays, and talked a little bit about how they function. I talked a little bit about, in bringing together various papers from the field, these impacts on human health, including fertility, potentially roles in dementia, cancer, and aging. here's a quick summary of my talk. This is the big take-home message that if you look at that top row, we use something called gamma H2AX as a way to visualize and show, hey, right here in a chromosome is a double-strand break. And on the left hand, when you add various things, which are the labels at the top, if you do nothing, the cells have a hazy green look to them. But when we added line 1, which is just mentioned here as L1, then you see lots and lots of spots develop. And that its ability to make these spots was dependent upon its endonuclease. So if you're endo minus, you make many fewer of these, although some are still there. If you get rid of the RT, which stands for reverse transcriptase, you also still get some, but very few are there. And then also, if you only have one of the two proteins that we'll talk about with this element, then you get some, but not as many. And in the lower hand side, the beautiful thing, this is called a comet assay. And so you look in the middle where it says L1, these make comet tails. And these comet tails are saying the DNA is being fragmented, there are double-strand breaks being present, and if you only have the vector, or you don't have the endonuclease, you don't see those tails. And so this was one of my primary publications, and all I'm going to summarize for today. And then this was the summary I gave, which is when we think about this element, this line 1, which has two proteins, an ORF1 and an ORF2, and these different individual activities and functions that these have, that these can interact with DNA, like you see in the diagram there. They're trying to insert themselves, but sometimes, their aim is to insert themselves and to replicate, but sometimes they don't do this very well. And so my work was showing that they just make a break in DNA. And so if you have in this next diagram under the light, when you have a double-strand break in cells, then there are genes that reply to that. And one of the genes you might recognize here is something called BRCA1. Anybody want to hazard a guess to what BRCA1 is known for? There have been big patent arguments and other stuff about it. Yeah, breast cancer. And so we know it's a breast cancer, that when it's gone, you're more susceptible to breast cancer because you have issues with repairing your DNA. And so the big question really was, when we think about the activity of these retrotransposons, the question is, how much do these contribute to a premature aging disease, like we see here on the lower hand right side, or this little painted bunch of blobs that you see? And this is an example of what's called chromosome painting, where normally you have chromosomes that look like they're either brown or they're red, but then sometimes now they've switched parts. That's called a translocation, and now they're a mosaic of different colors. And so I'm going to just, for the purpose of revisiting what we've learned since 2020, I'm just going to burn through some recent research since then. All you need to do, if you read the title of the slide, you look at the pretty picture, which I'll give a little description, read the green text, you should get the main gist of what each paper is saying. And we're just going to give the gist of the interesting research that has continued since 2020. So in the first one, talk about some work that is from Kathleen Burns, that L1 double-strand breaks drive mutagenesis in cancer. And the picture on the left is showing in their model cancer cell system that if you have L1 and then start expressing it, it just jumps around all these different places within the cell and lands on different chromosomes. The red triangles are representing a novel insertion that they found on, again, that chromosome depicted by the hash mark, black and white. And so the big body of work, though, is that they also showed that there are lots of times where chromosomes are rearranging, just like I showed you in that previous diagram where you're getting mosaics, but they were sequencing whole genomes, whole cells, and showing that the activity of the double-strand breaks are causing lots of things to rearrange and change and create this very typical cancer phenotype that you see of chromosomes going haywire and being relocated and messed up, as you see in ongoing cancers and as a driver of cancer. And so the green text just shows that, again, they are showing that alterations can arise from double-strand breaks, and this is coming from line 1. So a driver and an issue, and perhaps even an initiator of cancer, which fits in with a lot of research that has been done with line retrotransposons and cancer. The next one is an interesting model organism study, which is talking about how transposons can drive aging in nematodes. So nematodes are a wonderful little eukaryotic multicellular system that people work with in the lab. They're relatively easy to work with because they have short lifespans and they're easy to take care of. And so the diagram on the left, this is what's called, basically a survival curve. And the idea is that when they inhibit, when they keep transposons from being active, and that's what you have in the red lines, the fact that the red lines are to the right of the blue lines, then that is an indication of more longevity. Because what's being measured here is the percent that is surviving on any given day. So as the red extends out, more survival, and they're showing this by inhibiting the activity of transposons that are native. Again, the native transposons that C. elegans has in its genome. They also showed by the flip side that there are genes known to suppress transposons that are natural to all organisms. These are known as pewees, and that when they add more pewee to the mix, it also helps with that survival. There again has also been a variety of papers in humans, mice, and whatnot that have indicated this association of the expression of line 1 to things related to aging phenotypes. Okay, and the next slide. This was a cute one of, again, looking at another model organism, one that's used a lot for Parkinson's research, is the fruit fly, or Drosophila. And this one has a little more detail to the pretty pictures, but this one does one thing that's really, really neat, is that one, they mutated a gene, which we'll just call polyQ right now, but it's important for neuronal development. If you remove it, you have more problems with neuronal development. And that's what you see in the top-hand slide, is that, sorry, the very top chart in the diagram, looking at bad phenotypes for different versions of flies. And the red one is when you mutate that gene to the defective form. So you get a lot more bad-looking neurons. And then the blue and yellow are both adding reverse transcriptase inhibitors to those fruit flies and showing that it alleviates the bad cells. You look much more like a wild-type fruit fly only by inhibiting retrotransposon activity. And again, just to be clear, there isn't really any normal retrotransposon or retrotranscription activity that you have in normal cells. And you're seeing the same thing in the next two charts, that you knock out, again, the third column in each one is the mutated version without any treatment, and you have lots more problems in these. But then when you add in the far right chart, the far right bar graph, if you add a reverse transcriptase inhibitor, then suddenly you're alleviating all of the bad phenotypes, much more like the normal, which are the two left bar charts. And so, again, there's quite a number of interesting research going on with reverse transcriptase inhibitors and aging, as well as a variety of dementias, and there even is a human clinical trial that is, well, there are a couple. There's one hosted at University of Rochester, another one hosted, I think, at a Texas university. So, again, these are supporting evidence that the activity of these is driving problems with neuronal development in older age. All right, and then the last one, I mentioned fertility. And one of the interesting little things, when you think organismally about retrotransposons, we normally do as much as we can to keep them off or to limit their activity. And one of the few places where we have a limited amount of control over their expressing is in primordial germ cells, or in this development cycle from early reproductive cell stem cells to the point where they're becoming gametes. And so this work is showing, this is in mice, that if you eliminate a DNA repair gene, that ends up being bad for germ cells. And so, again, they're not using gamma H2X, they're using something called 53BP, 53BP1 as their indicator. And when they have more expression, when they knock out the DNA repair gene, they're seeing more double-strand breaks. That's what you see in the red. And then in that left-hand chart, again, they're doing something, again, very interesting with that. The KO is short for knockout. They remove the gene in those cells. And then the 3TC is the name of the reverse transcriptase inhibitor that they're adding. And so you'll notice that both for males and females, the knockout has higher levels of double-strand breaks present. And then when you add reverse transcriptase inhibitors, those all start to go away a whole lot more, much closer to the wild type. And so again, this is kind of this nice, elegant way of showing that there's this interplay between DNA repair genes, and then this is the problematic element that's going on in these cells has something to do with reverse transcription. So in summary, since 2020, there have been a lot of new papers looking at line 1 or other transposable elements driving genome instability, and this has been done in a variety of model organisms or in tissue culture, human cells, all sorts of the standard biology things we do in the lab. There's a lot more data on the connection between transposable element expression increasing in studied cells to ultracell phenotypes, and these expression elements are either measuring it directly or looking at something known as the methylation, the on-off pattern of the promoters of the things that drive their expression. And I think what we're seeing over again is that there's just this more convincing connections to cancer, aging, neuropathies, and fertility for all of the things I just mentioned above of genome instability, double-strand breaks, expression of these elements. And it's interesting to see some fairly clean suppression of the pathologies when you use reverse transcriptase inhibitors. Now, what's still lacking, and there's been a little bit of work on this, but not a lot, is trying to find an inhibitor that specifically targets the endonuclease, again, separate from the reverse transcriptase in these elements, which would be a nice solidifying factor for really narrowing down what the primary cause of some of this is. And that's because, having reverse transcriptase and cDNA, or again, complementary or RNA that becomes DNA, can cause other phenotypes like inflammation, right? We have a way of trying to fight off retroviruses, things that affect us all the time. And so that's something that could be driving some of these phenotypes. And then again, as I will remind people that next week, in 2 weeks, I guess 2 Mondays from now, they'll start announcing Nobel Prizes. But just to point out, I will not actually be waiting by the phone. So leaving a few minutes for questions or anything. I'll go back and double check the local chat if there are any questions, but let me know if there's any comments or questions you have about this work. Oh, so Miss asked an interesting question, which is, there's some promising research by increasing chromosomal disruption in cancer cells, and that's true. It's complicated because too much genomic instability will even kill a cancer cell. And so the trick is to do a combination really of two things, is that sometimes all of this activity that's messing up genome stability in a cancer cell, if you can actually inhibit some of these repair mechanisms that allow them to keep surviving, that ends up actually being a very powerful way, a very nice targeted way to deal with cancer cells, a very nice targeted chemotherapy. So maybe that's a talk I'll give in the future, because I find that to be rather fascinating. Yeah, prostate cancer is, of course, a big problem. It's one of these things that if you live long enough, there's a high likelihood you'll have some issues with the prostate. Not necessarily cancer, but some can also be precancerous. I'm actually not familiar with what Keytruda is, Roy, so I'll have to look that up and get back to you. Phil asked an interesting question. What about AI? From a biological experimentalist point of view, AI is really pretty powerful at helping you summarize information, sometimes finding interesting patterns. There are very specific ways of using it in drug or therapeutic target development. And so, yeah, it's definitely something there. To get into the specifics, we'll kind of see what plays out as actually being very powerful compared to how people's rational thought can help. I think they play a complementary role. Take take the things that AI does really well and complement that with the things that scientific brains do very well, and you actually have a very powerful combination. Yeah, and that's, I think, one thing Miss points out, that you'll have a high likelihood of having cancerous cells in your prostate, but in fact, it's not always worth, again, what you define as cancer, you don't necessarily have to treat it. There are lots of times where those do not necessarily become a health concern. But again, always talk to your doctor. Double check with your doctor and the most current information from a specialist on those types of things. All right, well, with that, Delia, are you ready for your part?
Yes. Thank you, Stephen. Yes, I am. Yes.
All right, cool. I'll go off speak.
Okay. So I'm glad to see everybody here today. This is the third time we have had a formal presentation up here. Well, actually fourth, I think. This is I did two here in 2020, and one at my my home garden in Neufreistadt. So we'll spend 10 minutes here, and then we will go over to check out the garden. So can everybody hear me first off? Okay.
Yes.
We're good then. So today I want to be connecting the dots and giving some updates on what has happened in research since 2020 in all of these areas. So the first thing I want to do is, when we were here before, we had, I focused on flood and fire. We didn't really focus on drought. There's been a whole lot more research on drought since then, in the last 5 years. So take a minute, stand up, please walk around. Tell me what you notice, what your guesses are, what you see, what you might know about drought conditions. Yeah, things are looking dry. So what does that mean? No green leaves, or very few. And the few that are green are a dried green. That's true. Dead trees. Yeah, that's the thing, dead roots. Yeah, the soil is cracked. It's very dried out, hard and dried out. And if you walk over here, you'll see more things. Dust, ashes. Yeah, the underground fires, the fires that catch the roots, tend to last a long time. And so even if there is some rain, there isn't enough to put out some of the fires. And then this happens in the temperate areas. It also happens in the tundra areas, where some of the wildfires in Russia have been burning for a couple of years. Yeah. Notice here you find a lot of dead stalks. This was a cornfield. So not only do you not have rain, what else do you have over here? What was this? Sis, what are you standing in? And Tag, what are you standing in? Yeah, this was a stream. This was a stream. It's dried up. That's a stream bed. All the plants around it are dead. And yeah, this is one of the problems. That since there was no surface water left, people have been pulling water from the ground. So the aquifers are getting dried out. And this is part of the really critical new research. So, and this research was done, and I'm going to walk us over to these two big pictures here. They're a little hard to see because the background of the originals is black, and it's just black and red. But this is a 22-year research project. And it was done using the data from the observational data from NASA's Gravity Recovery and Climate Experiment, or GRACE satellites. And that measures the change in mass of the Earth. And so this research has been applied to water. And they found something that surprised everybody, that they are able to estimate the water content and the change in water content. Yeah, mass of the Earth. Yeah, yeah. And particularly it's not just the total mass, but it is the location of mass. And that's the shift that is happening. So that if you look at the map on the left-hand side and find where you live in the RL. Now it's not just mass distribution near the surface. It goes deeper than that. It doesn't go all the way to the center, but it goes much deeper than just surface, because it's showing in shifts. What do you see where you live on the map that shows the data from 2003 to 2013 on the left-hand side? The black is where there was water. The red is where there's a lot less water. But there's more water, rather. So I'm lucky. I live in the northeast of the United States. So the northeast of the United States and some of the northeast of Canada is more black. Some of the lower areas of Canada were more black. And if you look at South America, you see that the Amazon rainforest in the top middle of South America was black. Yeah. So then if you look over to the right-hand side, find where you live there. Yeah, and I encourage people to go back and look at the old videos, and I will put the links back into chat so that you can easily do that, because there's nothing that is no longer valid in any of those. It's just that there's been an increase in research since then. So if you look at the right-hand side, yeah, what they have discovered now that they didn't realize before is that these are swaths of entire regions. And you look at Mexico, for instance. So Mexico is that little tail that hangs down in North America towards South America. Much of the food for the United States and a bunch of food for Canada comes from Mexico. Vegetables and fruits particularly. Much of the meat, particularly beef, comes from Brazil. Those areas are now red. They are now in drought. And these are regions, swaths across the entire globe where the food growing areas are in danger, really in danger. So I think of this particular area, and I'm going to put another piece in here that is gives you a whole lot of other links here, or a few other links about this particular research. But if you think of, I think of this particular area, this build, as we're standing on it, as, is anybody, are people familiar with Charles Dickens' story, A Christmas Carol? Yeah, because this to me is like the Christmas of the future if Scrooge didn't change. And this is the way that we are moving if we don't change. This is a long-term trend. It has gone on. Yeah, it's the ghost of the future if we continue down the path that we're going on now. But it's not hopeless. It's not hopeless because we can make the changes that we need to make. The question is, how are we going to do that? How are we going to do that? What has to happen in order for us to make changes so this is not the future that we get? This is not the future that we live in. So here, I'm going to put now, because we're halfway through here of this little bit, a link to my pasture and where my small garden plot is there, because I would like everybody to come over there, and then we'll talk more about some of the future, some of the solutions, some of the things that we can do that are going to mitigate and ameliorate some of these particular issues, okay? And please come back here and explore more. But let's end this. So, this is my home in Second Life. When we were here before, it was winter time. It's now going toward the end of harvest. And the garden, rather than being covered in snow, it has still a whole bunch of vegetables. There are fruit trees. And some harvest is already put in the barn so that I will have some food for winter. Now, I do not grow all of my food in SL, nor do I grow all of my food in RL. But I do grow some, and I grow a lot of the herbs that I use. And there are a bunch of things that I have learned in doing that come really dovetail well with some of the recent research. And that is about soil microbes. When I did this talk in 2020, we did not know a whole lot about the extent that microbes in the soil were critically important to the immune systems of plants and to all of the ways that we need to have a healthy, diverse set of biomes. So if you're not familiar at all with soil biomes and all, here is a chart right in front of me that'll give you some idea of what we're talking about and some of the microbes, because it's more than just the kinds of fungi, for instance, that we now know are both the paths for communicating among plants, but also the paths for knitting soil together. And what we know now also that we didn't know before is that we have the soil microbes have a huge, huge effect on the carbon sequestration and climate change in that sense. And most of the models that are talking about climate change do not have that built in. So that's one of the areas that they are now trying to shore up and make sure that they have a better handle on what the climate change actually is. So there are a handful of things that connect this to the drought area. One is that soil microbes, when the soil is healthy, there's a diversity of soil microbes, and they're pretty drought resistant. They may be slowed down. You might not have the things growing that you would normally grow, but it doesn't necessarily kill them off right away. What does kill them off, though, is the intense application of pesticides in our industrial agriculture that is across the globe today. And the monoculture of growing corn and soybeans particularly, that changes the soil biome. Instead of being a diverse mix that is healthy, is when you have the monoculture, it also has an effect on the soil biomes, that they become much more homogeneous and much less resilient. So when you have the kill-off, and when you have that of the biomes, then you don't have the critters that really knit the soil together. Then you're much more likely to have things like dust bowls. So what I've tried to do here, and what I've tried to do in that other solid life as well, is not only learn about the biomes, the microbiomes, and the little critters that live in the soil, but also do what I can to ameliorate the condition of my soil. So here you'll see that I have a compost bin that usually is full of a goat or two. And I do that in the RL as well. So even the times that I have lived in an apartment and didn't have a garden of my own, I would still do the recycling of vegetable wastes and make sure that I contributed that to add to the soil and have put it back somewhere. Because if we take it out of Mexico, for instance, and deposit it in the U.S., we are virtually extracting the soil from the health of the soil from Mexico, but it's not put back anywhere, and it runs off into the ocean, which is not a good thing because that adds nutrients where they don't belong. But anyway, so this is an example on a very small scale of what we can do to ensure that we don't end up with that future that nobody wants. We're really compressed in time today, so I'm not going to go further. I certainly talk more about this or do a whole presentation at some time, if people would like. Yeah, the soil loss, it's not just in North America, though, Stephen. It is very much across the growing areas of the entire globe. So it's in Europe, it's in China, it's in South America. And while you're here, since this is harvest time, please, please, please walk over and take yourself a little bit of, or a lot, of freshly pressed apple cider and some goat and sheep cheese that are on the table right in front of the barn entrance. Yeah, when the Amazon tag, as we cut any of the tropical forests, they do not grow back ever as the same kind of tropical forest, because this is those plants and trees have very shallow roots, and once they are removed and disturbed, that has a huge effect on the soil. And so you're not going to grow back those kinds of trees on the very dried out soil. But as important is that the density of the tropical rainforests affect the weather, particularly not only the Amazon, but in the Democratic Republic of Congo and in Indonesia. These are huge rainforest areas, and they affect the weather and particularly affect the rain patterns across the globe. So that when you have cut down the trees in the Amazon, for instance, you have eliminated the sources of future rain for that area. And you're right, Tag, that will, if we continue this, it will turn into savanna. And a lot of people don't have the space to do actual gardening outside their homes. But you can still do some, even as a window box or small containers inside, because you learn a whole lot about plants and how they're grown, how they grow in doing that. So I really, really encourage you to begin to learn some of this and find out, where's your food grown? The stuff that you eat all the time, where does it come from? And are there better sources for you to continue to buy, even at supermarkets? Buy organic, buy ones that are non-GMO, pesticide-free, because those are available in a lot of supermarkets. And although none of our individual choices are going to make a huge difference, if we are doing this across the globe, it definitely makes a difference. Yeah, organic is sometimes more expensive, not always, because you can often get that at farmers markets. It's not necessarily more expensive. But one of the reasons that organic is more expensive is that industrial agriculture, particularly in the United States and Europe, is subsidized. Organic agriculture is small farm agriculture for the most part, and it is much, much less subsidized. So we're paying tax money to encourage the kinds of food growing that hurt us. We do. We pay farmers not to grow. That's not always a bad thing, though, Neuro. We don't want to overgrow something that isn't needed. That was the source of that originally. And some of the farmers who are in that kind of a program and are being paid for not growing are turning, particularly in the Midwest and West of the United States, are turning those lots into prairie land, which is a very good thing. Sis, I'll get you the link to some of the papers on that. And this is a ProPublica summary, but it's not the originals. But it's primarily the loss of groundwater, the loss of aquifers that they're measuring here. And as an example, just quickly, there is a huge aquifer that goes through the western part of the United States. So it's called the Ogallala Aquifer. It goes underneath 8 states. But they're not all the same. So in the northern part of the aquifer, it's a lot deeper, and it tends to be more solid rock. It tends to be more granite or rock that has harder structure. In Texas, though, it tends to be more karst, which is a porous limestone. So yeah, that's where you're going to get in Texas, across the lower part of that aquifer, and also in Florida, you're going to get sinkholes, because that's where the water is drained out to an unsustainable degree, and then the aquifer collapses. It's not a strong structure. And then that part of the aquifer can never refill because the space is gone. Yeah, yeah. In the shallow areas, and Texas, for instance, has had laws that permit somebody to buy a small plot of land, put a water drill there, suck out the water, say 1,000 feet down, and sell it. So what that's done in a handful of towns so far in Texas, and this has actually happened, is that the farmers and the townspeople who live in that area and have had wells that are 200 feet down, 400 feet down even, have dry wells, and they can't drill further because there's no water for them. And so there are abandoned towns now in Texas, small towns to be for sure, but they have had to abandon the towns because there is no source of water for them anymore. This has also happened with a new build in Arizona. Yeah, Nestle has done that in California. Yeah. Yeah. So it's up to us, though, to change these things. It's up to us to get ourselves educated, to make small choices in how we buy things, what we buy, to talk to other people in our communities about whether you are using your resources in your community in a way that is sustainable. Because we can do this. This is not impossible. Not yet, anyway. Yeah, that's what I was saying, Tag. Yeah, there is a learning curve. Start small. Start with something in pots that you like. Say you like tomatoes. Next summer, grow a couple of tomato plants in a pot. Experiment. If you can't do that, put something on a windowsill. Learn a little bit about it. Yeah, I have encouraged in my RL, and I'm trying to do that here also, is encourage the pollinators. So in my RL yard, I have, for the most part, native trees and shrubs and grasses. And it makes a huge difference in the pollinators that you attract. If all you can grow, all you have the space for, is something that looks ornamental, yeah, lettuce is easy, that's very true, and it grows pretty quickly. But say you like basil, for instance. Basil grows quickly. Plant some basil in a pot, or cilantro if you like that.